Leading a startup team, whether you're delivering a sugar rush, stocking coffee, or getting a regular delivery of snacks, Office Depot has solutions that fit every startup culture, from getting those first business cards and stationery to ordering fleece pullovers with your new logo. To learn how Office Depot and the California Technology Council have partnered to bring you savings on all of these startup essentials and more, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report. By using data from routine testing about the genetic evolution of the HIV/AIDS virus in the Canadian province of British Columbia, researchers have shown they're able to identify emerging outbreaks of the disease in near real time and intervene to control its spread. The first of its kind system is the focus of a study in an April edition of the Lancet HIV. We spoke to Art Poon, Associate Research Scientist for the British Columbia Center for Excellence in HIV/AIDS, and lead author of the study, about the monitoring system, the impact it's had, and whether such an approach is transferable to other regions and other types of disease outbreaks. Art, thanks for joining us.
Thank you very much.
We're going to discuss your recent article in The Lancet about a case study of real-time monitoring of HIV hotspots. Perhaps you can begin by explaining what the BC Center for Excellence for HIV/AIDS does.
Okay, so the BC Center for Excellence in HIV/AIDS is a provincial agency. It provides all clinical services and treatments to all residents of BC, British Columbia, living with HIV. And that comes at no cost to the patients. That's fully subsidized. In addition, the BC Center is a world-class center for multidisciplinary research. It's probably best known for a couple of things. One is being the kind of epicenter for treatment as prevention, the concept that getting individuals onto antiretroviral treatment as early as possible makes them less likely to transmit the virus, and so there's a prevention benefit to antiretroviral treatments. The other thing is that the BC Center is responsible for the scientific evaluation for pretty much the first sanctioned safe injection site in North America, Insight, which has had a tremendous benefit for the well-being and clinical outcomes of in the injection drug user population in BC. There are many other research arms going on at the BC Center, but there are just too many to name here, so I've just picked out those two.
HIV is a virus that evolves rapidly. You routinely test for resistance to antiretroviral therapies through genotyping. How can genotyping of the virus give an indication of the relationship one case of HIV has to another?
So you're absolutely right. The HIV evolves extremely rapidly. So an infection will become genetically unique in a matter of weeks or months because of the constant mutations that are appearing in the virus population within each patient. When we see two infections that retain genetic similarity, that tells us that those infections are related by one or more recent transmissions, that there hasn't been that much time since those infections, those populations, descended from their common ancestor. Now, this doesn't tell us anything about specific transmission events. It doesn't tell us that A infected B or B infected A, or it can't dismiss other individuals like C infecting A and B. And we don't want to get at that level of transmission. What we're interested in is the variation in rates of transmission, trying to identify uh, outbreaks of recent transmission at the population level.
There's a lot of HIV testing that goes on, but for the most part, I take it these tests are done in isolation, that they're used to diagnose an individual patient rather than aggregated to provide public health monitoring. What's the opportunity from a public health perspective to look at this data more holistically and in real time?
So, looking at these data at a population level has two key benefits from a public health perspective. The first is that it allows us to understand where the most vulnerable groups are to onward HIV transmission in the province and be able to get that in near real time because the HIV genotypes are already being routinely collected as part of standard of care. They're already accumulating at the BC Centre. So this is a way of making secondary use of that resource. The second benefit is by looking at these data at a population level, it maintains a high level of data privacy. Because we're focusing on the population, we're not trying to get at an individual level. We're not, in fact, we're designing the system so that you can't get at an individual level of transmission. We are just trying to characterize the patterns of transmission at the level of populations.
Well, let's walk through the study, which used a database from the BC Center for Excellence for HIV/AIDS. What exactly did you do, and what data were you looking at?
So this the system that we described in this study is built on a secure Oracle database at the BC Center for Excellence. It operates through what's called a restricted view, and so the system can only see certain anonymized and non-identifiable variables in the database. And so it operates on that restricted part of the database. The system is made up of a number of scripts, Python scripts that I've written that automate the database transactions, process the HIV genotype data that it retrieves from the database, and then reconstructs phylogeny. That's a tree that represents how all of these different infections are related through common ancestors. Now, this is operating on roughly 33,000 sequences that represent almost 9,000 people in BC living with HIV. In order to do that, the system communicates with a high-performance computing cluster at the BC Center through a secure encrypted channel. That's how that we're able to regenerate the evolutionary relationships among all of these infections in a short period of time and be able to keep things as close to real time as possible. The last step of the system is that it uses the information from this phylogenetic analysis to populate document templates. This is a combination of a templating language called Jinja and document typesetting language known as LaTeX. So these report templates are what we use to communicate results to our partners in public health.
And what was the result?
And the result was that, well, first of all, we were able to generate network diagrams that visualize what's going on with respect to the epidemic and with respect to localized outbreaks as they may occur. The limiting step was figuring out how to take these network diagrams and translate them to public health. So it's work with people in public health and understand how they view these diagrams and how they want to extract information from them. So embedding these diagrams into reports that were regularly being disseminated to public health, our partners in public health, medical health officers, was an evolving collaboration that required a fair amount of time, and really more time than it took to implement the system. The key result that we talk about in the paper was an outbreak that is known as Cluster 55. This was an outbreak of about 11 new cases over a 3-month period. What was alarming about this was, first of all, this was a cluster of originally roughly 25 cases, so that was a very rapid expansion. And second, 8 of those 11 cases had transmitted drug resistance. And that means that those individuals had a virus that already carried a mutation that made the virus resistant to an entire class of drugs. That would rule out first-line antiretroviral therapies for those individuals and is really clinically significant, not only clinically significant, but also significant from a public health standpoint. As a result, that precipitated public health follow-up on this population. That action was followed by a period of reduced accumulation of new cases in the cluster, and also a marked reduction in the occurrence of transmitted drug resistance.
You talk about the fact that the data was anonymized. Were there ethical or privacy concerns that served as any kind of a hindrance to doing this?
So the ethical and privacy concerns of implementing this system were probably foremost in our minds throughout the whole development period and remained that way. Before I wrote a line of code to implement the system, the BC Center spent years laying out the groundwork for the ethical and privacy framework for the protection of individuals, working with the Ministry of Health in BC. It wasn't until that was in place before we did any work in implementing the system. The system is designed to maintain the same high level of data privacy that exists in the BC Center laboratory. The idea is that we're not changing any of the anonymization and data privacy that's already in place. Any of the data that the system operates on are securely erased from the hard drives the instant that the analysis is complete, for example.
Well, what were you able to do differently once you identified an HIV cluster?
So what really changes with being able to identify an HIV cluster is being able to find vulnerable populations in the province. So one of the key objectives of treatment as prevention is the idea of getting to zero, that is, zero incidence of new HIV cases. In order to do that in a population where we've already been able to reduce the number of new cases by rolling out treatment as prevention, the challenge is finding where the new transmissions are occurring in a large population. So a cluster identified by this system can represent a vulnerable group that can most benefit from HIV prevention services in near real time. We don't have to wait until that outbreak has reached a point where it becomes really obvious. We can look to the system and try to get at that outbreak sooner and prevent new cases from accumulating as soon as possible.
The benefits of this approach, would you say it's able to improve outcomes or reduce the spread of the disease?
So I mentioned the Cluster 55 public health action before, and that we found was associated with a marked reduction in transmitted drug resistance. There were new cases that eventually appeared over time in that cluster, but the majority of those cases did not carry that resistance mutation. There was no transmitted drug resistance. And so that suggests that the public health follow-up that was supported by the monitoring system was at least partially successful in preventing that onward transmission of drug resistance. We don't know if the system had a direct benefit because we don't do some sort of case control study where we withhold public health follow-up in a similar cluster if we saw a similar outbreak and wanted to see what would happen in the absence of that public health action. So we're still working out and trying to understand how the system may improve outcomes and benefit the population. Because this is the first of its kind, we can't look to any other place for guidance. And the evaluation of our system as really something that's groundbreaking is the sort of key objective for our further research, trying to understand and quantify, do these interventions have a measurable impact on HIV prevention?
How easily replicated is this model?
This model, in one aspect, is easy to replicate because routine HIV genotyping is standard of care. And so there are a lot of settings around the world where HIV genotypes are constantly accumulating at these clinical research centers. As a matter of fact, there is a lot of interest in implementing a system very much like ours, especially in the United States. And that for that reason, some of our collaborators at the U.S. Center for Disease Control, the CDC, have asked for our source code, in which we agreed to share with them. So the system that we've implemented in BC is kind of providing a groundwork for building up similar systems around the world. On the other hand, it's not as easy because one thing that we really benefit from in British Columbia is highly centralized healthcare. All of the clinical and genotype data, demographic data, that's all integrated into one centralized database. And so it's sort of more difficult for other groups to implement a similar system, for example, in the United States, where you don't have that centralization of providing care.
There are some unique aspects to HIV/AIDS, and you've got the genotype testing that's being done regularly. Is this applicable to other diseases? Do you see this type of monitoring being used to control other types of outbreaks?
This is absolutely applicable to other diseases. The prerequisites are that you need routine genotyping, so you have to have data that you can act on. And you need a pathogen that evolves rapidly. That covers many viruses, and it also covers bacteria when you bring in whole genome sequencing that's possible, efficiently possible with new next-generation sequence technologies. We're presently looking at translating this system to hepatitis C virus. Now, HCV has it's been brought to the forefront of the clinical world, not only because it's highly prevalent in the baby boomer population as well as injection drug users, but also because of the new generation of drugs, the direct-acting antivirals, that while effective are also prohibitively expensive, and that may be an important consideration for targeting clinical services to core transmission groups in the population. That means that you need to have the kind of insight into your epidemic that the monitoring system can confer in order to be the most cost-effective.
Art Poon, Associate Research Scientist for the British Columbia Center for Excellence in HIV/AIDS. Art, thanks so much for your time today.
Thank you very much for talking to me.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.